Okay, welcome everybody. This is our first podcast dealing with Chapter 3, where we're going to explore the cell, which is the smallest unit of living matter. So we'll start out with just an overview of what we would call a typical cell. And you're seeing really everything we're going to talk about in this one diagram. The nucleus will start at the center of the cell, contains a genetic material which is DNA, and this DNA, in order to reduce damage and retain integrity, is wound around little tiny proteins that are called histones. There's also within the nucleus an area known as the nucleolus, which is the site of synthesis of ribosomal RNA, which is a structural component of ribosomes, which, as we're going to find out, are the protein synthesizers. They use the material that's contained in the messenger RNA molecule in order to build the new protein. The nucleus is surrounded by a phospholipid bilayer membrane which contains nuclear pores which allow certain substances to get in and out of the nucleus. And the reason that this is important is because as the cell calls on the nucleus in order to pull up instructions for how to build particular proteins, the way that it does that is to actually send specific enzymes into the nucleus through these nuclear pores so that they can locate the genes responsible for those proteins, open them up, transcribe them into messenger RNA, and then that messenger RNA will be transported out into the cytoplasm, which is everything between the nuclear membrane and the cell membrane, which is this outer layer here. The cytoplasm has all kinds of interesting stuff in it. Just to take a quick tour, this labyrinth of sacs is known as the rough endoplasmic reticulum. It's called that because it's studded with ribosomes, which are the protein synthesizers. Once the messenger RNA molecule leaves the nucleus through the nuclear pore, the ribosomes will then bind to that message and read it 3 nucleotides at a time in information blocks known as codons, and use that information to synthesize the nascent protein, the polypeptide chain that will eventually be the active protein. Once the ribosome reaches the end of the messenger RNA, what will happen is that it will release that protein and it'll be sent into the rough endoplasmic reticulum, and as it moves through the rough ER, it will begin to fold into its native conformation, which is the shape in which it's going to do its final job inside the cell. Eventually what will happen is that the proteins will bud off the end of the rough endoplasmic reticulum, fuse with the Golgi apparatus. The Golgi apparatus serves as a protein distribution warehouse. It receives vesicles from the rough ER and then tags them with molecules such as polysaccharides to indicate where they should be loaded once they're sent off to their final destination into the cell. And once that occurs, once the Golgi vesicle buds off of the face that's furthest from the rough ER, what will happen is that that vesicle will eventually be transported to the location where it's going to do its job in the cell. That vesicle will then release its protein components and then will eventually move back to the Golgi apparatus to repeat the process. And the way that these vesicles know how to move around is due to the cytoskeletal components that are inside the cell, which are made up of microtubules, intermediate filaments, and microfilaments. And you could think of them all as pulleys or cables or train tracks that help guide a lot of these other organelles around inside the cell cytoplasm. Another very important cellular organelle, you can see them, they look like little sausages all throughout the cytoplasm, are the mitochondria. These are the organelles in the cell that are responsible for making the bulk of the ATP, which is the chemical energy currency of the cell that it uses immediately to drive all other cellular processes. The centrioles are responsible for the synthesis and the breakdown of microtubules, which are particularly important in the process of mitosis, where duplicated genetic information is evenly divided between 2 brand new cells and distributed to the 2 brand new cells at the end of mitosis and cytokinesis. Other organelles that are important include the lysosomes. You could think of them as the stomach of the cell, and what they do is help recycle worn out organelles and also break down components that the cell engulfs in a process known as phagocytosis to take inside itself, either for the purposes of feeding itself or for the purpose of destroying things that invade the body, such as pathogens. There are a series of diseases known as lysosomal storage diseases that can adversely affect cell function to the point where waste products build up to such a degree inside the cell that the cell can no longer perform life functions. The smooth endoplasmic reticulum is so named because it does not have ribosomes on its surface. It's the site of phospholipid synthesis, and it's also the site of detoxification reactions. Other important organelles include peroxisomes, which are detoxifiers inside the cell. What they do is destroy free radicals, which are electron-poor species that can run around inside living systems, stealing electrons from chemical bonds, resulting in the destruction of those molecules and eventually the destruction of the cell. And you can, of course, see the microtubules, the microfilaments, and the intermediate filaments running throughout the cytoplasm, which is a gel-like substance which isn't, I mean, it appears as if it's a homogeneous mixture to the naked eye, but really it's full of enzymes and electrolytes, vitamins, cofactors, of course, tons and tons of cytoskeletal components, all doing their work, not necessarily visible to the naked eye unless they're properly stained. The outer periphery of the cell is covered by something known as the cell membrane, which is another phospholipid bilayer. Incidentally, we should point out that all of these organelles are surrounded by phospholipid bilayers, and the purpose of this bilayer is to contain the cell components so that they don't wander away. It also serves as a semi-permeable membrane for the cell, meaning that certain things can come in and certain things can go out. It's not simply an open door for anything to leave or enter whenever it sees fit. Another important function of the cell membrane is that it contains embedded proteins that allow the cell to be recognized as belonging to the human body. There's also a wide variety of receptors, channels, and pumps that regulate the cell's internal environment. They're constantly utilizing energy in order to maintain homeostasis for the cell itself. The same can be said for the membranes that surround a lot of the cellular organelles. It's very important, for instance, to keep certain sets of chemical reactions confined to a particular space inside the cell, because if they were allowed to work anywhere they liked inside the cell, it would result in cellular chaos. As an example, the lysosome contains enzymes that are very good at breaking down protein and polysaccharides and all manner of organic molecules. They're contained within inside the lysosomal membrane. If they were allowed to escape, they would begin the process of digesting and destroying the cell. And in fact, when the cell gets ready to die, that is one of the things that happens, is that the lysosomes begin to actually disgorge their enzymatic components and begin to break the cell down, and that ends the cell's effective lifespan. So we'll explore each of these organelles in more detail as we walk through the cell piece by piece. So let's begin with a discussion of the plasma membrane, the cytoplasm that contains the organelles, and the nucleus. So remember that the cell is the smallest unit of living matter. So it is able to perform a lot of the characteristics that we associate with living things. It's able to reproduce, it's able to grow and differentiate and metabolize, it's able to respond to its environment. All of these things are part and parcel of a normal cell's lifespan. It performs metabolism, which allows it to take the energy that's locked in the chemical bonds in the fuel molecules that it takes in and turn that into usable energy for the cell in the form of ATP. It's also involved in anabolic processes, where it builds large molecules up from smaller components so that the cell can use that to perform its function within the context of the human body. It's also important in communication. Cells produce and receive electrical and chemical signals from the outside world. These signals operate primarily through the action of ligands binding to receptors or certain channels or gates, either opening or closing depending on certain conditions in the cell membrane at that particular point in time. The communication function of the cell is seen most easily in nerve cells and in muscle cells. These cells have the ability to respond to electrical signals by producing a voltage change in the membrane, and that causes them to perform a certain activity. Generally, in the case of a muscle cell, it's to contract. In the case of a nerve cell, it's to send or receive electrical information and relay it down a pathway, either to the central nervous system or from the central nervous system to an effector. But cells can also communicate with each other chemically. Hormones are an example of chemical communication in cells, which are small molecules that are excreted into the bloodstream and carried throughout the body via the circulatory system, and then they bind to cells that have the appropriate receptor for that particular hormone, and then that causes them to perform an activity designated by that hormonal secretion, and as a result create conditions within the human body that cause that original signal to be shut down, and that's what we call negative feedback. They're also important in reproduction and inheritance. Each cell contains DNA, which contains all the information that controls how the cell looks and acts and the kinds of molecules that the cell synthesizes. Some cells are specialized to form what we call gametes, which are important in sexual reproduction. A gamete has half the amount of genetic material as a normal body cell. A normal human body cell has 46 chromosomes, and a sperm or an egg cell will contain 23. And it's in the process of fertilization, when the sperm and the egg meet each other for the first time, that we restore the diploid condition, and we now begin the process of making a new human being. The plasma membrane is the outer boundary of the cell. It separates the intracellular and the extracellular environment, and it's made up of a phospholipid bilayer. Remember, in the last chapter we talked about phospholipids as being amphiphatic molecules, meaning that there's one part of the molecule that's polar and likes to associate very readily with water, and the other portion of the molecule is hydrophobic and likes to associate with other hydrophobic molecules. And so in an aqueous solution, one of the stable arrangements of these molecules is to form a double-layered membrane with the hydrophobic tails projecting inside the phospholipid bilayer and the polar heads being exposed either to the water in the extracellular environment or to the cytoplasm in the case of the intracellular environment. Other important roles for the plasma membrane include the regulation of intracellular and extracellular ion concentration through the action of pumps and channels. The outside of the membrane in a typical living cell is positively charged compared to the inside of the membrane, and this is due to a couple of factors. One being the action of a very important pump in the cell membrane of every living cell called the sodium-potassium ATPase, which pumps 3 sodium cations out of the cell for every 2 potassium cations it pumps in, and it uses an ATP molecule every time it does that. And also the fact that the cell membrane is differentially leaky to sodium and potassium. It's more leaky to certain cations than others, and that's responsible for the charge difference inside and outside the cell. And the significance of this establishment of the separation of charge across the cell membrane is that it serves as a form of stored energy that the cell can use a lot like a battery in order to drive a lot of processes that are critical to maintaining homeostasis in the cell. The glycocalyx is a combination of carbs and lipids, which we call glycolipids, as well as carbs and proteins, which we call glycoproteins, which are on the outer surface. The purpose of these glycolipids and glycoproteins, in part, is recognition of that cell as belonging within the context of the human body, so that you can think of them kind of as ID tags. They can also serve as adhesion molecules. They allow cells to stick to certain things. The whole membrane itself you can think of as a two-dimensional moving network of molecules. These let's draw a picture here. Pick a pen color. Let's pick, oh, I don't know, yellow. That'll show up. These phospholipids, in the context of the bilayer, can move laterally. They can move back and forth or left and right, depending on the molecules that are around them. as can a lot of the membrane proteins, the pumps and the channels and the receptors and so forth. And so the membrane is a dynamic changing thing, which is important for the cell because the cell needs to regulate its composition as its environment outside the cell changes from moment to moment. So that's the skinny on the cell membrane. Other obvious functions for the cell membrane include keeping all the organelles in one place and, of course, serving as a semi-permeable barrier. Okay, phospholipids and cholesterol predominate the cell membrane. The phospholipids form the phospholipid bilayer, and we've already described the arrangement of these molecules. Cholesterol is used as a way to mediate how much the membrane moves around. So if you have a lot of cholesterol inserted into the membrane, there will be less movement of the phospholipids. If you have less cholesterol, there will be more movement of the phospholipids. And the purpose of this is so that the cell can regulate whether it wants certain things to move around and aggregate or not. Remember that cholesterol is a lipid itself. As a result, it is hydrophobic, and so it is stable within the hydrophobic tail portion of the phospholipid bilayer. This fluid nature allows distribution of molecules within the membrane. Phospholipids are automatically reassembled if the membrane becomes damaged, and these membranes can also fuse with each other, and this is important in processes such as endocytosis and exocytosis, which are two forms of what we're going to address, active transport that the cell can use to move things either into or out of it, and it uses ATP as an energy source. Okay, let's talk a little bit about these membrane proteins and where they're found, what they look like, and kind of what they do. Integral or intrinsic membrane proteins extend deeply into the membrane, and they often extend from one surface to the next. They can form channels that go all the way through the membrane, and this provides a portion of what we call the semi-permeability of the membrane. This is what allows certain things through and other things not. Peripheral or extrinsic proteins are attached either to the extracellular half of the phospholipid bilayer and protrude into the extracellular environment, or the interior portion of the phospholipid bilayer where they protrude into the cytoplasm. And their function depends on their shape and their chemical characteristics. They can serve as markers, attachment sites, channels, receptors, enzymes, pumps, or carriers. Okay, all are possible roles for these membrane proteins. So you don't have to think of the cell membrane as simply nothing more than a phospholipid bilayer. Glycoproteins and glycolipids allow the cell to identify itself as belonging to that particular organism. They serve a very important role in immunity. They also allow the recognition of the sperm and egg cell when they fuse in the act of fertilization. These glycoproteins and glycolipids are a very important component in that particular phenomenon, and they're also vital in intercellular communication in the context of molecules such as integrins, where cells can attach to other cells and by attaching send a chemical signal to the cell to which it attaches that can be relayed to another cell or can be shared between the two cells. And so you can see what they sort of look like. They have a protein component, and then they have a polysaccharide component as well. You probably know them better in their role in the immune system as antigens, okay, the ID tags that identify these proteins as serving that role within the body. Cadherins attach cells to other cells, while integrins are integral proteins that can attach to an extracellular molecule. They sometimes allow communication due to contact with intracellular molecules, so they can have both a structural role but also a communication role. Okay, so you can see how these attachment proteins, these integrins, can recognize a particular extracellular matrix protein, and this allows for the potential for the cell to remain functionally attached to that molecule, but can also serve a signaling role. The integrins can be attached to cytoskeletal components or can be attached to other enzymes that can trigger a cascade inside the cell that will make it perform a particular action, okay, perform some sort of function. Now, transport proteins do exactly what you think. They move things in and out of the cell. The hydrophilic region is going to face inward towards the portion of the transport protein that's responsible for moving that particular solute in or out of the cell, and the charge in the interior of this protein will determine what kind of molecules can pass, as will the shape of that channel. These transport proteins include things like channel proteins, carrier proteins, and ATP-powered pumps, and you could just think of them as doorways to let certain things in and out of the cell. And they can function in any of a variety of different ways. They can function passively, meaning that they don't require an energy input in order to drive the movement of one particular solute from the outside of the cell to the inside of the cell. They just operate through diffusion. As long as the door is open, the molecule will diffuse from where it's most concentrated to where it's least concentrated. Or they can be active, in which case they use ATP to drive the movement of molecules either with or against their concentration gradients, depending on the needs of the cell at that particular moment. Okay, so think of them as doorways, as passageways. Non-gated ion channels are always open, and as you can guess from the name, what they are doorways for ions to move in or out of the cell. They're responsible for the permeability of the plasma membrane to ions, even when the membrane is theoretically at rest. While gated ion channels are open or shut depending on conditions within the cell membrane at that particular point in time. A ligand-gated channel will open up depending on whether or not it has bound its ligand. What is a ligand? It's a small molecule that attaches to the binding site of the gated channel and pops the door open. Voltage-gated channels will open or shut depending on the difference in charge between the inside and the outside of the membrane around that particular channel protein. So depending on the voltage, the gate may open or the gate may remain shut. And you can see examples of these channels here. Here's a potassium leak channel. As long as it is open, what will happen is that the potassium will naturally diffuse through the leak channel from the inside of the membrane to the outside of the membrane, which is with its concentration gradient. This is a gated sodium channel, as another example of a membrane channel protein. In this case, the gated sodium channel is closed, and here is a ligand-gated sodium channel which has bound its ligand, has opened up, and now has allowed the sodium to flow down its concentration gradient from the outside of the cell to the inside of the cell. Okay, so those are all examples of a form of passive transport. They're all working with the concentration gradient to move the molecule either in or out of the cell. Carrier proteins, also known as transporters, are integral proteins that will move solute molecules from one side of the membrane to the other. Sometimes these are ions, sometimes these are other types of molecules. They have specific binding sites for their particular substrate. The protein will then change shape to transport the ion or molecule from either the inside of the cell to the outside or vice versa, depending on how the pump is, or how the channel protein, the carrier protein, is operating. And it'll resume its original shape after the transport. And again, these can be powered by ATP, or they can be passive proteins that work with the concentration gradient of the solute that we're considering. And they come in several flavors. Uniporters move a single molecule, a single solute. Symporters move two or more solutes in the same direction at the same time. And antiporters move two molecules in opposite directions at the same time. And again, these can be either powered or they can be passive examples of transport proteins, meaning that they work, again, with the concentration gradient to do their job. When we talk about active transport, we're talking about transport that's powered by the universal energy currency in living systems, which is ATP. And this allows the cell, as a result of using this energy source, to move the solute molecule in any direction it sees fit, either with or against its concentration gradient. The rate of transport is going to depend on the concentration of the substrate as well as the amount of ATP that the cell can use to perform the task. Okay, now receptor proteins are another example of an integral membrane protein. They can be either pure proteins or glycoproteins, which means they have a carbohydrate moiety, and they'll have a site that's on the extracellular surface which acts as a receptor for a particular ligand. And a ligand can be any type of molecule, could be a protein, could be an ion, could be a modified amino acid, could be any number of potential signals which can attach to the specific ligand activated receptor protein and cause it to become activated. The example you're looking at here is a particular type of receptor protein called a G protein. What happens with a G protein is that a chemical signal will activate it. This in turn will trigger the release of GDP by the next protein downstream in this cascade, which is intimately associated with this ligand activated receptor. This protein will then bind to GTP, that will activate it, and then it in turn will activate another protein either in the cytoplasm or perhaps in the membrane as well. And what will happen as a result is that the signal cascade at each step will grow in magnitude. If we were to diagram this, we could show you sort of how this works. If you imagine this is the membrane, okay, the double-layered membrane, this being our receptor, and then we bind a ligand. Let's give the ligand a color. We'll color it green. The ligand binds here. This in turn activates, oops, sorry about that. This in turn will activate a downstream protein. So let's indicate this in black. So an inactive protein, which we'll call 0, it becomes activated. We'll call that 0 star. Okay. In the case of the G protein, in case of a receptor protein that's associated with a G protein, this would be the G protein here. Okay, so let's put G protein. Sorry, apologize for my legibility here. G protein, PR. And then this G protein will in turn activate another protein. We just call it protein X. And protein X goes from an inactive to an active form. And at every step in this cascade, what happens is that an activated protein, whether it be the G protein here or protein X here or the next protein in the cascade, which we could call protein Y, they can inactivate more than one downstream target. The result is that the signal grows in magnitude geometrically at each stage in the cascade. And the wonderful thing about this setup is that it's easy to turn off because all you have to do is stop the first or second points in the cascade, and there are enzymes that are already resident in the cytoplasm that will deactivate the proteins that have been activated as a result of this cascade. The result is that we can turn it off very quickly. Note that the ligands attach only to cells that have that specific receptor, so they'll pass by any cell that does not have the appropriate binding site on the cell surface to which the chemical signal can attach. An example of how this works is if we look at hormones. Hormones are secreted by glands that are present throughout the body. The hormones are chemical signals that get sent into the bloodstream through active transport and are carried all over the body, but the only target tissues are going to be those that have the receptor for that particular hormone, and when they bind it, what will happen is a series of cascades, just as we've diagrammed here, that will ultimately result in the cell doing something. Okay, make sure and review this material. It will be covered on the next PRS quiz, and I will see everybody in class.